Hi everybody, welcome back to biology class. So as I explained in that introductory video, I'm going to publish these slide narrations every now and then, just sort of like highlighting some key points. This first one is not particularly too in depth. It's just sort of like to sort of get the ball rolling, let you see how this is going to go. So we are pretty much done with cellular respiration. We are done with glycolysis, with the Krebs cycle, with oxidative phosphorylation, at least until the final anyway. So we do want to move on to genetics. I know in class I said I wanted to move on to the cell cycle next. However, I think with this distance learning opportunity, it sort of makes more sense to go into genetics first, and then we can go into cell cycle once we hopefully meet up again as a class. So if we are going to talk about genetics, though, we're going to have to talk about the father of genetics, and that, of course, is Father Gregor Mendel. So Mendel was an Austrian monk. He was born in the Silesian region of the Austrian Empire, what is now modern-day Czech Republic. And he actually loved school. He loved learning. He loved science. And he went to school himself to be a high school teacher. So he traveled off to the city one day and took the exam to be a high school teacher, and he failed the exam. So he then traveled to Vienna, Austria, and studied physics for a few years under a physicist there by the name of Christian Doppler. And the name Doppler probably sounds familiar to you. The Doppler effect is named after him, and meteorologists used Doppler radar, which is also named after him as well. So he studied under a very, well-renowned, well-respected physicist for a number of years. And he went back to the city again to take the teaching exam, but for a second time, he failed. It's not that he was a bad student. It's not that he didn't understand the material. He was just a very anxious test taker. Just couldn't quite, get over it. So he then went on to the monastery and joined the religious life instead. So he always still had this, this passion, this zeal for science. And so he continued doing experiments even while, serving as a monk in a monastery. And his experiments, of course, centered around this idea of inheritance. And what he kind of found out was genetics is all about inheritance. It's all about these sorts of packets of information that he referred to as factors that get passed on from one generation to another. So he was able to actually notice and catalog that these things that were happening were not just random, but they were the result of a certain pattern that came about. And so in his day, of course, they didn't have the molecular understanding that we do today. They did not know about things like chromosomes or DNA. Certainly didn't know about genes. So he used the term factors to explain what it was he was seeing. A lot of people in his time thought that traits were blended. So you take your mom, you take your dad, you put them together, and you are just a representation of a blending of their traits. Sort of the same way you take red paint, blue paint, you put them together and you get purple paint. However, when you do that, you cannot get that original red or blue color back again. However, with traits, with genes, you do see those sorts of characteristics pop up yet again in subsequent generations. For example, there might be traits that your grandparents have that your parents do not have. However, maybe you do have those traits. So that could not be explained by blending. There must be something else going on here, and that something else must be located in these little discrete units, these factors that control inheritance. So here we can actually see Mendel holding a pea plant. This is at the monastery in Brno, Austria. And once again, that's part of the Czech Republic these days. But he actually did not start out by studying pea plants. He actually started by studying mice. However, the religious order did not really care much for him doing experiments with these live animals. And so they asked him to please choose something else to study instead. So he started studying bees. But the bees that he chose were very aggressive, unfortunately. And so even though he loved working with the bees, he got a lot of complaints from his fellow monks. He got a lot of complaints from visitors. And the abbot eventually told him he had to get rid of the bees. So he instead moved on to peas. And peas really, when you think about it, make a lot of sense. You can get a lot of generations in a very short amount of time. You could also control their breeding. So working with pea plants, proved to be very, very beneficial. So he was actually started off by using purebred plants. So if you wanted a plant with purple flowers, for example, he would take plants with purple flowers and breed them over and over and over again until all of the offspring had purple flowers. Then that would be considered a purebred plant. The same sort of thing with white flowered plants. He would breed them over and over and over again until every single plant produced white flowers. Then he would take a purebred purple flower plant and crossbreed it with a purebred white flower plant. So the purebred generation, that first generation was considered the P generation. And the first generation offspring, the crossbred, the hybrid, were considered what's called the F1 generation. So then he would take that F1 generation and crossbreed them to get an F2 generation. So it's kind of like, grandparent, parent, and child, except he was doing that with pea plants. So he controlled their breeding through a number of different ways. One way he did this was just placing a bag over the top of the flower so that if it was producing pollen, the pollen could not escape anywhere. It would just get trapped on the inside of the bag. Another way he did this, however, was by actually physically removing the male reproductive part of the flower. So the part that actually produces the pollen. He would just use scissors and cut off the stamen. So both the anther and the filament get cut off. In that case, there's no pollen that can be produced, and that plant cannot therefore pass along its genetic material. Another way you could control breeding, however, was to take like a paintbrush and scrape off some of the pollen grains from the stamen and brush them onto the pistil, the stigma of the female plant. And that way he was ensured the pollen that was being received by that female plant was by the plants that he had chosen. So using pea plants, he was actually able to sort of like control the parameters of experiment much better and still get, legitimate results from it. One thing he was doing, however, he was really looking for these sort of binary either or traits. So he wanted to start out with a purebred tall plant or a purebred short plant, a purebred, purple flower plant or a purebred white flower plant, and so on. And so as he did this, as he was looking for his binary traits, he was actually able to breed over 29,000 pea plants over a 14-year period. And he started noticing these patterns that kept popping up. No matter which trait it was he was studying, he kept seeing the same numbers come out. So whether it was the seed shape, round versus wrinkled, or maybe even the pod shape, inflated versus constricted, no matter which trait it was he was looking for, he kept seeing the same pattern show up in the F2 generation. And here we can see that much more clearly. So he's noticing that no matter what it is he's looking for, he's seeing about 75% of the time one particular trait shows up, and about 25% of the time the other trait shows up. The trait that showed up 75% of the time he can refer to as the dominant trait, and the one that only showed up 25% of the time he can refer to as the recessive trait. So there's always this sort of 3-to-1 ratio of traits that were popping up dominant to recessive in the F2 generation. So he's showing that there's not blending occurring, that there must be these sorts of discrete packets of information contained on these factors that he called them that would determine which trait would actually show up. And they did so at a very regular rate, regular and predictable rate. This isn't random. So he did publish his study, but within 35 years after his study was published, it had only been cited 3 times. So despite the fact that there was something happening here, despite the fact that maybe it wasn't quite entirely clear, it should have given, pause to a lot of scientists to say, hey, what's, going down? Everybody look what Mendel's found. Unfortunately, it was pretty much just ignored. If you didn't get that reference, maybe your parents or grandparents do. But it was, kind of like because of the title of his study, it was It referred to hybridization. And, we've had hybridizing plants for thousands of years. This wasn't something, hybridization itself wasn't something new. So people just look at the title of the study and move on and not really look at the information he actually discovered. So it wasn't until much, much later, long after he had died, unfortunately, that he started getting the recognition he deserved for his work. And also, it just goes to show you cannot, just as you cannot judge a book by its cover, you should not, judge a scientific study by its title. Go ahead, flip through the thing, look through it, and see what sort of information is held in there. Because what he discovered here really was revolutionary and groundbreaking. So for now, I think we'll, sort of pause it for the time being. Just so you know, like I said before, I'm going to pretty much do these every now and then. I'm only going to talk for 10 to 15 minutes at a time at most. I'm certainly not going to talk for 45 minutes. I don't want that. You don't want that, and nobody wants that. So just sort of like try to hit on some highlights, some key points. This particular presentation, not a lot here. It's just like sort of an intro, but we'll get more into the meat of this material as we go on. But I did want to just provide sort of like an understanding of what it is we're going to be doing and how this is going to look. So if you have questions, of course, please feel free to email me. I hope we're back together again soon, but for the time being, take care, be well.